Very much for the invitation, for hosting me. I arrived and, Madhav noticed that I was wearing a white shirt, so he brought me somewhere that was serving Indian curry for lunch, and so you may be able to see some of that. So I apologize that I don't look as spick and span as I was hoping to for you guys. But yeah, so it's wonderful to be here and to have the opportunity to tell you a bit about the kinds of ideas that we've been excited about in my group in the physics department over at MIT. So just kind of as a starting point, I think many of us across different scales are interested in this question of, how is it that different kinds of structure or function arises at one scale, maybe the level of a school of fish or a population or a cell, right? But how is it that these sorts of structures and behaviors, functions, arise from the interactions between the players at a smaller scale? Right now, of course, the answer to this question could be very different depending upon the system or depending upon the scale. But in my lab, we're often interested in this question at the level of populations and communities. Right? How is it that interactions between individuals in a population or between species in a community lead to interesting sort of dynamics? Right? So for example, at the level of fish, you have these cooperative populations that may help with avoiding something like predation. And interestingly, this occurs both at the level of the predator and the prey. Right? So the school of fish may help to avoid predation by the predators, but also you have kind of group behaviors at the level of the predator. Right? So dolphins, for example, engage in various kinds of group hunting behaviors that allow them to capture and eat more fish. Right? So in that case, as an example of the kinds of behaviors that arise, if you have these cooperative populations, then you have, for example, tipping points in populations and catastrophic collapse. And this is thought to occur in fisheries and in other systems. And this is the kind of dynamics that we are often interested in my group, right? So how is it that you have some sort of interaction at one scale that leads to maybe surprising things at another scale? And so in my group, we're often interested in these dynamics not only across different physical scales, but also across different time scales, right? For time scales that are less than a generation time, we're often thinking about behaviors. Right, so how is it you get the emergence of spatial patterns within, say, a population? Over timescales that are similar to the generation time, then you're really thinking about population dynamics or ecological dynamics. Right? So for example, you might think about how is it that a fishery may collapse in response to overfishing? And then finally, over longer timescales long compared to the generation time, you might think about the evolutionary dynamics of the population. So for example, if fishing is preferentially directed towards mature or large fish, then you may lead to you may get an evolution of the population towards earlier reproduction and spawning. Okay, so this are the kinds of dynamics that can take place across these different timescales. Now, for me as an experimentalist, these are all maybe interesting kinds of phenomena: pattern formation, tipping points, evolution. But these are difficult things to study, over the timescale of. Well, people always say over the timescale of a PhD, but for me, what was relevant was over the timescale of tenure, right? So similar timescale, and you want to be able to to study some of these phenomena and get some insight. So what we do in my group is largely study these sorts of phenomena or ideas, but using laboratory microcosms. So populations of bacteria or yeast or maybe worms, right? So experimentally tractable populations where the timescale here, the generation time, is something maybe reasonable. And it might be a few hours for our microbial populations. Okay. Now, within these microbial populations, if you look at them, indeed, they have many behaviors that are similar to what we were looking at with the fish. Right? You have the emergence of spatially structured or patterned populations. You have cooperative growth. You have evolutionary dynamics that are obviously very important for antibiotic resistance and other things. And also, you have tremendously complex communities. Right? So this is a false-colored EM image from human stool. And what you can see is that if you look close enough, it doesn't look so bad. Right? You have nice colors, pretty things. Right? But the key thing that you see, though, is that it's a tremendously diverse community. Now, the question in a lot of this field of ecology is, right, what is the origin of this diversity and then the functions that are associated with that diversity? What is the origin of that diversity? Okay. And I'll just mention that there are a number of theoretical ideas here. that point to some possible mechanisms. Okay? So, for example, there's thoughts that some sort of what you might call higher order interactions may be a significant contributor to the diversity that we see within a variety of different communities, both at the micro scale and at the macro scale. Right, so, for example, there was some recent nice work from Roy Kashoni showing that if you have maybe some degradation of toxins of one species by another species, this could be a contributor towards the diversity within a community. Similarly, at the macro scale, there's been a number of pieces of work pointing out that if you have one of these higher order interactions, again, where a third species is fundamentally changing the nature of the interaction between another pair, then this could be a way to get more diversity than what you might expect based on just the pairwise interactions between the species. All right, so just as a concrete question that we're going to be kind of focusing on today, I just wanted to mention something about our approach. Yeah, so I mentioned that we use laboratory microcosms. Now, the way that we often think about this is we're interested in the microbes as individuals, right, or as communities in and of themselves, but we also like our microbial populations and communities because we think that they're a powerful bridge between the theories from, say, mathematical ecology or other mathematical fields, and so bridge between the mathematics and the really complex communities that we're often maybe most interested in. For microbial populations, it might be, say, the human gut microbiome, but for other organisms, it might be the behavior of a fishery, right? But in both cases, what we think is that by working with these experimentally tractable populations in the lab, we maybe can explore the models in some ways and also influence the way that people think about the more complex communities. And of course, we ourselves are influenced by both. All right, so now just as, all right, so the human gut is too complex for me. So a lot of what we're going to be thinking about today is just the dynamics of very simple communities, such as here I'm going to show you some data from a 3-species community. So these are 3 different bacterial species, an Enterobacter and 2 different Pseudomonids. And what I'm going to show you is just what happens when you compete these 3 species in some different environmental conditions. All right, so over here, if you can see, all right, so this is something where we're looking at the fraction of this population as a function of time. And what you see is that it's the blue Pseudomonas veronii that ends up taking over the population over here on the left. I haven't yet specified what this is. I'll tell you more about it in a moment. All right, but this is just one environmental condition where PV wins, drives the other 2 extinct. Now what we're going to do is imagine, okay, we're going to change the environment. For now, you can just think about deterioration. Yes? Ecological. Oh, sorry, this is ecological timescale. So this is about 50 generations. Exactly, yep. Thank you. All right, so now, okay, we're going to vary the environment in some way. I'll tell you what it is later. Now we're going to look at kind of the other end of the spectrum, where over here what we see is now it's the Enterobacter that is the one that wins, outcompetes the other 2 species. And I'll note that throughout this entire range of environmental conditions, each of the 3 species could have survived on its own. Okay, so it's just highlighting, all right, so as you turn some experimental knob, you get a very different outcome. Okay. All right, so now we come over here and say, all right, so it looks very similar. Still, the red EA species is the one that wins. Maybe takes a little bit longer for the other 2 species to go extinct, but a qualitatively similar outcome. Okay. Coming over here, now what we're seeing is that here there's coexistence between 2 of the 3 species. So now the red and the purple species are able to coexist in this particular environment together, whereas the blue species is driven extinct. Okay. Coming over here, all right, now we've just gone a little bit to the right. Now what we're getting is coexistence, but now it's between the red and the blue species, the purple species that is driven extinct. Okay, continuing to march along in different conditions. Here again, there's coexistence between the 2, and indeed other experiments confirm that, so it doesn't keep on coming down. But interestingly, at this environmental condition, we don't always see coexistence between red and purple. Sometimes we see coexistence between red and blue. And it turns out that at this environmental condition, it's actually bistable. There's alternative stable states between where we always see that the red survives, but sometimes it survives with one partner and sometimes it survives with the other partner. Right? It depends upon the initial abundances of those two species. And indeed, you can see how the initial conditions are different in those two experiments. And there are other experiments you can do to repeat it. So it really is a bistable system at that particular environmental condition. Yeah, so this is all in well-mixed conditions. And I'm going to show what the experiments actually look like. But for now, I just want to highlight that you don't have to go to 100 species in order to start looking at data and being like, gee, this is kind of complicated, right? So this is just one experimental knob where you can see that there's a lot of different outcomes. And the way that we like to think about this or simplify things is to use these things that we call subway plots. So yeah, I don't know what your guys' elevated train maps look like, but this is kind of like what it looks like in Boston, right? The idea here is that the presence of the color or the line tells you about whether that species survives in competition with those other species at that environmental condition. So over here, only blue survives. Over here, only red survives. Here, it's red and purple, red and blue. And then the two lines being separated like that is telling us that there's bistability at that environmental condition. Yeah? So generally, no. Right? So we actually have done one experiment where we had coexistence of 5 different species in a particular environment, and then we were interested in the coevolutionary dynamics. So we allowed them to go for about 350 generations. So some evolution definitely taking place, and we did this in replicates. So we had 96 different replicates where we started with 96 different versions of these ecosystems, and what we found is that in all 96 different versions of those communities, all 5 of those species survived in all 96 different replicates, but in different proportions. So the evolution then went in different places, but the forces for coexistence in that, and I think other experiments, are actually quite strong. So if there is coexistence, then even with some evolution, the coexistence is preserved under even some evolutionary timescales. Oh, yeah, it continues to vary. So what we normally see is that over these short ecological timescales, it's sort of at a stable equilibrium. But then if you go to evolutionary timescales, then the things diverge. Although in that experiment, what we saw is that there was kind of clustering in the space, in abundant space, right? So 30% of the communities went to abundant space over here, 20% went over here, right? And that's the kind of thing you can only see by doing this, like looking at many replicates. But yeah, okay, that's a whole other story. But I guess the statement is that the coexistence, when it occurs, is often quite strong. Yes? Okay. Yeah. Yeah, okay, so these are all very important questions. So throughout this talk, when I say extinction, what I mean is that it's below the detection of the experiment that we were using, which is typically about 1%. Right? So it's very much possible that the species is surviving in low abundance, and in some cases we can explicitly demonstrate that. We haven't done that in this particular system, so I don't know the answer to your particular question. But for now, extinction is 1%. In some other cases, we can see though that we can get the reemergence of things later. And so yeah, but for now, just say below detection. The equivalent thereof, yeah. So this data was all from plating. Yeah. All right, so what I want you to get from here is just that a variety of things can happen as you change experimental conditions. This is all actually in the same media. Turns out this is just changing how much we dilute by each day. Okay, but it's highlighting that things are already complicated with 3 species. So what we want to do is at least try to understand this, and then it may or may not say anything about the human gut. But we want to start somewhere. Are there any other questions about where we are right now? Okay, so now I'll tell you what I'll tell you about. Okay, so I'm just going to start by trying to understand pairwise competition. What are the typical things that happen? And then I'm going to ask, if we know about the outcome of pairwise competition, what can we say about three-species competition? Or in general, we're interested in multi-species competition. And what we're going to find is that actually the pairwise outcomes typically do a pretty good job of predicting the 3 or 4 or 5 species outcomes. In particular, for example, that previous 3-species diagram that I showed you, it's essentially entirely predicted by the pairwise outcomes. All right, I'll show you what I mean by that, but that's where we're going. And in order to get there, though, well, I'm going to have to first tell you about, all right, well, how is it that these environmental communities change under stress? In particular, under increased mortality, the pairwise outcomes shift in predictable ways. And that's what allows us to kind of go from the pairs to at least the trios. Okay, so just to be clear about now what the experiments are looking like, what we're going to be doing is we're going to be competing 2, 3, 4, but basic for now, pairs of species in liquid media. It's going to be well mixed for now, but it doesn't actually make that much of a, nothing I'm going to tell you, none of the qualitative conclusions really make a difference whether we shake or whether we just leave it static. But for now, you can imagine it's well mixed. So we're trying to prevent any spatial structure. Start as simple as possible. Okay. Now, what we do is we just add these 2 species. We allow them to grow and compete doing whatever it is they do. Their resource competition, toxin production, whatever it is they're doing to each other. And we're going to just perform dilution here every either 24 or 48 hours into fresh media with fresh nutrients and everything else. And then they'll grow back, compete. And we'll do this 5 to 7 times. So we go about, say, 60 generations. Okay? With the idea being that we want to determine the ecological outcome of the competition before there's significant time for new mutants to arise and for evolution to do its thing. Right, so our goal is to try to focus on ecology first, and then evolution we can try to solve the next time I come. Okay? All right, and in this initial experiment that was led by Jonathan Friedman, who's now has his own group at the Hebrew University of Jerusalem, he just took 8 different soil bacteria, with E. coli is just shown here as an out group. So 8 different soil bacteria. He did all pairwise competitions, and then from that he asked, can I predict the outcome of all the trio competition? Yeah. Yes. Yeah. So this was all just from plating. So he counted, 1 to 200 different colonies, and they all have different colony morphologies. Actually, yeah. So very low tech, which for a variety of reasons I like more than sequencing, but that's a whole other discussion. I'm happy to talk about it later. Okay, so that's the idea, and this is getting to this conceptual question, right? So there's a practical question. Oh, if you want to know who's going to survive, can we do it? But there's also a conceptual question of how important are higher order interactions in these microbial communities? I think it's not obvious. So among these 8 species, there were very simple competitive outcomes. And indeed, he really just saw 2 things. Sometimes he saw competitive exclusion, where one species drove the other one extinct. Right? And what you can see is that, and this is some experimental data, where it didn't matter where he started the competition, the outcome was always the same, where this PP outcompeted PV. And we're just going to illustrate that with an arrow going from the PV towards the PP. So from the loser to the winner. It's like the flow lines of the fraction. The other thing that he often saw was coexistence, where here, still PP, but now it's against a different species, PA, where you see, again, it doesn't matter where he started the competition. He gets stable coexistence at some equilibrium fraction. Okay? The way that we're going to display that is just by 2 arrows coming together to that equilibrium point. And among these 8 species, these are the only qualitative things that he saw, either competitive exclusion or coexistence. Very simple outcomes. Later, I'll show you some cases where we get bistability, and that's going to be important for leading to the bistability in that other trio. But in this study, they were all just competitive exclusion or coexistence, right? And I think that that's nice because then you can just plot for these 8 species the different experimentally measured outcomes. What you can see is that there's quite a lot of coexistence, but with a significant minority of of competitive exclusion. In addition, you can see that some of the species are more fit than others, in the sense that, for example, PP up here, it outcompetes 3 other species in red, and it coexists with the other 4. Right? So this PP is not driven extinct by any other species in this collection. Okay. Right, now there are many things that you might want to do with this. You might want to analyze the network motifs, and we have done some of that. But for now, we just want to focus on this basic question of from the pairwise outcomes here that we've measured, can we predict the trio outcomes? All right, so the way that we might so coming back to the cartoon that we had before, right, how many of you guys have seen Alien v Predator? All right, Madhav and I have a lot in common here. All right, it's a surprisingly good movie, right? Yes. No, I mean really, right? Okay, and I'm going to tell you a little bit of the plot, which is very important to the story. So if you're going to watch it, you can cover your ears, all right? But basically, in this movie, indeed, as if you were a boy growing up in the 80s, like a common thing that you would argue with your friends is which one was more powerful, right, between the aliens and the predators, okay? Because they were different franchises, and of course the answer is obvious because the predator is much more powerful, and that's partly because of this shoulder-mounted like laser rifle thing that he has, okay? So the predator can outcompete the alien in pairwise competition, okay? However, in this movie, there's a third species that's present, which is humans. Okay, so these people, they show up and they sort of accidentally come across this weapon before the predator can get it, and that kind of complicates the whole situation. Okay, now I'm not going to tell you what happens, but yes. Which one is E. coli? Yeah, all right. Well, I'll let you decide at the end of the day. Yeah. Right. The point is just that even if you know the pairwise outcomes, it's not obvious what's going to happen when you have 3 species. That's what you're supposed to get from this. But it is a formal movie recommendation as well. Yes. So it could be anything, right? And we've done a lot of work to figure out what it is. So it's probably not so much toxin production here, although these are real species doing whatever it is they do. So there could be some. It's probably mostly via resource competition. But the important statement is that it's not just growth rate. There are a lot of cases where the slower growing species outcompetes the faster growing species, and then you can argue about exactly what it is. But it's certainly not as simple as just the growth rate. All right, so this is a particular trio. 3 species where on the edges is our experimentally measured outcome of pairwise competition. All right, so this Serratia loses to the Enterobacter, whereas the other 2 pairs display coexistence. Right, so we can look at this triangle from one standpoint as just being a drawing of what we measured in the pairwise competitions. But the nice thing about these triangles is that we can also use them as the simplex corresponding to the competition of the 3 species. Right? The idea of that is just that any point in the interior corresponds to some relative fraction of the 3 species. If you're right in the middle, that corresponds to equal abundance of each 3. And then as you get closer to each of the species names, that corresponds to more of that species being present. All right. So the question here is, well, given that, all right, if we want to predict the trio outcome from the pairwise outcome, well, maybe we should vote. We talked about active learning and so forth. All right. So, and this is also a way of checking to make sure that you guys understand what we mean by these arrows. Okay. So everybody get 25 seconds to make a guess of what's going to happen if we compete the 3 species together. All right, and what we're going to do is we're going to do it by pointing. All right, so you can point whatever you want. If you think SM is going to win, you point down there. EA, PA, coexistence. You can point right at me to say that you think it's going to coexist to the 3. You guys understand All right, ready? 3, 2, 1. All right, so we got a lot of Travolta going on. All right, so indeed, the prediction is that we're going to end up here, that with coexistence of EA and PA, right? And would somebody like to say why they voted for that? Flow, right? They're flow lines, and this is part of what's nice about, right? Is that the only allowed flow lines given this triangle? Yeah, exactly, right? So I think that the logic is maybe, well, in pairwise competition, the EA outcompetes the SM. So maybe that's what's going to happen in the trio, right? And this is basically the heart of our proposed community assembly rules, which is just that you're only going to be able to coexist in the multi-species community if you're not outcompeted by anybody that's surviving in pairwise competition, right? It does not have to be true, and we'll see some flow lines where actually other things happen. So it's, yeah, but the flow lines are at least a good way of thinking about it. Yeah, right, okay, right. So, but that's over evolutionary timescales, right? So the nice thing is in ecology, it should all be kind of some well-behaved flow lines, is my claim. Okay, of course, the whole point of making predictions is that you go and you make experimental measurements, right? So what we did is we just started 2 replicate communities down here dominated by SM, 2 up here dominated by EA, 2 down here dominated by PA, and then we just looked to see what happened in these multi-species communities as we compete. Okay? Right, our prediction is just it's going to survive the trio if and only if it can survive in the pairwise competition. Okay? And you can generalize this to more species, but for now that works. All right, and when you go and do the measurement, it's exactly what you guys all predicted, right? You can see that, and it doesn't matter where we started. In all 6 of these replicate competitions, what happened is that the serratia goes extinct, and you end up with coexistence of the EA and the PA. And very nicely, you see that the fraction of those 2 was the same as what we had originally measured when we did the pairwise competition before. Okay, that's encouraging because I think there's often this idea that when you're thinking about ecology, it's like, anything can happen. None of the measurements are going to be reproducible, and we should just give up. And I think that that's an overly fatalistic view, and that if, at least for these microbial populations, where you can have, you can have a constant environment and reproduce, then ideally you should be able to see something like this. This is not earth-shattering. It's exactly what we maybe would have expected would have happened. And also, I just picked a trio. I could have just picked one where that prediction worked. So we're going to have to do more experiments, but at least this is a way of getting started. Yeah. Yeah, so here it's based on CFUs, so the colony forming units, basically the number. And that's what's being plotted based on these starting conditions. So it was like, I think 90%, 5%, 5% was our starting. Yeah. Okay, so in this particular trio, a simple assembly rule worked. But then, if we want to say something more general, then we have to make lots more measurements. So, we didn't actually, it didn't, so Jonathan went and he measured all 8 choose 3 trios, all combinations of the 3, and went and did that same experiment where he started in different places and then made measurements of where they went. And you can see, what happens, right? Where they kind of like cluster here, just PP. So I'd say overall it looks pretty good. There may be some emergent bistability. Okay, so there's interesting things that you can go and study. But the key takeaway from all this is just that assembly rule that I mentioned, that we mentioned before, although very, very simple, does, I'd say, a surprisingly good job of predicting which species are going to survive in the context of the trio competition. Right? In particular, you get about 90% prediction ability, which is kind of an A- or B+, depending upon your grading scheme. Right? The cases where it fails is split roughly equally between false survival and false extinction. So it's saying that this very simple rule does, I'd say, a pretty good job of predicting whether a species is going to survive in the context of the trio competition. Now, of course, the cases where it fails, it's not just that our measurement didn't work. It's like it really fails. But that's good because then it points towards maybe some interesting things that we can go study in terms of things like toxin degradation and so forth. And I do want to show, though, a particular trio that fails, but is still consistent with the flow lines. And actually, I'd say everything is consistent with the flow lines. Okay, and then I'll make the point that it's not necessarily, though, due to higher order interaction. Okay, all right. So this is another trio, and the first thing you should note is that, okay, so 3 different species, but the outer edges are the same as what we had before. In the sense, on the left, there's a case of exclusion, and then the other 2 pairs display coexistence. Okay? Yet, now there's something I think that's kind of surprising that happens, which is that it's the species PP, which was the species that I told you was the most fit of those 8 species, that is actually the one that goes extinct. And it goes extinct in all 6 replicates. It's not like, oh, we just once didn't measure it. You can see it didn't matter where we started. We could even start dominated by PP, and then we still end up down at the bottom where PP goes extinct. Right? So this is, I think, interesting on a number of levels. Right? So it's first, I think, telling us that, that you can have some surprising things. Just because a species can't be excluded by anybody in pairwise competition, it may be that it can be excluded by a combination of other species, and this is the simplest instantiation of that. And this is maybe reminiscent of some things that we think may be happening in the human gut. Right? So like a C. diff infection is thought to be an alternative stable state that's very difficult to cure. Right? So even with antibiotic treatment, often the C. diff comes right back. Right? However, what's been observed is that a fecal transplant often actually succeeds in reversing that state. Right? So it's a complex community that together can displace the C. diff. And this is the simplest version of that. Just 2 species together are able to displace PP, where neither of them could have done so on their own. And you can see that it is consistent with the flow lines around the edges. It's just that here, this fixed point, whereas before, normally it's stable. In this one, it's an unstable fixed point. Right? So you can kind of come right through. All right, so there are multiple ways that you can have consistent flow lines. And indeed, actually, this triangle, there are 4 consistent flow lines corresponding to the 2 times 2 different stabilities of these fixed points. And actually, we see 1 instantiation of each of those 4. Right? So there were something like 25 times where we saw this triangle, and maybe like 21 times it did the thing that we expected. And then once we saw each of the other things. Yeah. Yeah. Yeah, we could have. Yeah, so you want to know if it's really invasible. No, I'm thinking. Oh, you're wondering this could be stable if we get closer, right? Or not if it's. Oh, I see. Yeah. There's something weird with it. Yeah, sorry. So there has to be like some like. Yeah. What does there have to be? Yeah. Right. So there has to be like some dash line that curves them over. Yeah. And in a lot of these cases, we have we have gone where we just add a very little bit of one to demonstrate that, but this is actually just one of the 56 different trios. So yeah, it's not that it's special, but it's kind of interesting, I would say. Yeah. Okay. Although this was a case of this failed prediction, it actually is not proof of a so-called higher order interaction, because actually this is one of the allowed outcomes even within this Lotka-Volterra model that I'll tell you about here in a moment, which contains only pairwise interactions. Okay. So although this is somehow a surprising outcome, it can arise even in a purely pairwise model. All right, so now we have this kind of claim or this belief that in a given environment, if you tell me the pairwise outcomes, I can make some sort of prediction of what happens when there are 3 species. Okay. Now the question is, what can we do? What can we say in general about changing environments? Right? So this is a project that Clara Breu, a PhD student in my group, has led. And she wanted to know, okay, well, if you have a complex community like this, and you put it under stress, antibiotics, diarrhea, something else, right? What will the community look like? The abundances, which species are going to survive and which ones will not? Okay. Again, complicated problem. So we're just going to start by asking, can we actually say anything about what happens with a pair? And then, since pairs seem to do a pretty good job of at least 3 species, see if we can build up from there. So this is the bottom-up approach that I was referring to. All right. So in general, and we want to take a step back, before we just saw exclusion and coexistence. In general, there are other things that, of course, can happen. Right, so I told you before, okay, so exclusion or dominance is the other thing that we often call it. coexistence, but often we do see bistability, where at the final state, only one or the other species is surviving, but which species survives depends upon the initial abundances of the species. Okay? And this is like what you might get in a lot of antagonistic interactions. Between two armies, the larger army, you'd say, oh, will likely win. Okay? So if you have some sort of antagonism like that, then you can naturally get something that looks like this. And I would say something like 95% of all pairs that we've done look like one of these three things: either dominance / competitive exclusion, coexistence, or bistability. Okay? And then 5% of the time, we see something else. And something else could contain many, many different things. Right? So sometimes it's just what looks like there's rapid evolution, so just these curves go crazy. Right? And that's like, okay, well, I'm not sure what we're going to do with that. Sometimes we actually do see bistability with one of the states being coexistence. And in at least one case, we have seen oscillations where the two strains, and this was a case of two antibiotic-resistant strains that together could grow in the presence of two antibiotics. Right? So they were breaking down each antibiotic, allowed the mutualism to survive, and that actually did oscillate. There's a limit cycle oscillation. It's very interesting from a mathematical dynamical systems thing, but I'm not going to say anything more about that, except just that there are other things that can happen, but almost always we get one of these three things. And that's nice because those are the three qualitative outcomes that you get from this most simple model of interspecies competition, this Lotka-Volterra competition model. All right. Now, the basic idea is that depending on the strength of the interspecies inhibition, you can either have, if they inhibit each other strongly, you get bistability. They inhibit each other weakly, you get coexistence. And then you can have exclusion or dominance depending on which way you're looking at it. Okay? The model is very simple. Right? So it looks something like this, where over here on the left, this is the per capita growth rate of the population size n. There's some maximum growth rate that is going to decrease based on some self-inhibition, just that the species will always use up resources eventually, as well as some inhibition by the competitor. Right? So this is maybe just the simplest model you could write down or imagine to capture the basic idea that the species are interacting in some way. And for now, we'll assume that they're competitive, so the alphas are positive. All right, so as a starting point, the model is at least vaguely reasonable because it predicts these basic outcomes that we at least see experimentally. Now the question is, what can we do with this model that would allow us to make some more interesting prediction about what changes as we turn some experimental knob? And what we want to do in this case is ask, what happens if we impose a mortality that is experienced equally by both species? And so now what we're going to do is we're going to call one of the species the fast grower and the other species the slow grower without loss of generality, just because we're going to assume that RF is greater than RS. Okay, so there's a fast grower and a slow grower. Now, in addition, we've added this term delta that is some sort of mortality that is the same for the two species. Okay, so we're assuming that there's some deterioration in the environment. We're going to impose a dilution, but in general, some deterioration of the environment that is experienced equally by the two species. All right, now, at least in this model, the nice thing is that the prediction is very, very simple. And that's because this delta term can actually be just absorbed into the other parameters of the model. And what we get is that over here, if we can start out here where the slow grower outcompetes the fast grower because it's a strong competitor. Its alpha for the other guy is large. Okay? Now, if we start out over here, as we increase this mortality rate, the delta, you travel down and to the right at a 45-degree angle. Very simple prediction, where eventually you can get the fast grower to outcompete the slow grower. But along the way, you'll cross either a region of coexistence or a region of bistability, but not both. And what I like about this is that it's a qualitative prediction that could be robust to the fact that this model is not actually a good description of any real species. Right? So for example, here, the assumption is that you saturate logistically, and we can measure that. We see it's not saturating in this simple way. The interaction is not going to be this simple thing, right? So we know that the model is not accurate in that sense, but the question is, does it contain enough truth to guide our experiments and what actually is going to happen? Yes? That's right. To first order, no. Yeah, so we've done both. I mean, there's also the theory that you can do discrete and you can do the experiments continuous. We've done both. Yeah, so it doesn't. It doesn't really matter. Once you include a lag time, then you can get more subtle things in a variety of ways. But at least in the simple model, actually, the only thing that matters is time averaged mortality rate in this model. And we've demonstrated that experimentally as well, actually. Yeah. Okay, so now we come back to the experiments that I was telling you about, right? So we're just growing competing. We do this daily dilution, right? So what we're going to do is we're going to vary how much we dilute by, and that's going to change our mortality rate that is experienced by the species. Okay? So a larger dilution corresponds to a higher effective mortality rate. So a very simple experimental manipulation. All right. So we can just look at a couple species. All right, so this is the density as a function of time of just two species in some environment. What you see is that EA in red is a faster grower than PV. Okay? That's fine. However, when you compete them, what you see is that actually the fast grower is the one that goes extinct. All right, so the fast grower is driven extinct by the slow grower. Right? It can happen, right? So what we wanted to ask though is, all right, well, what happens as we vary the dilution rate? And in particular, can we travel down and to the right as we impose a larger dilution? Okay. So here's some data. Fraction goes to 0. And the basic answer is yes. Right? So there's a region in here at intermediate dilutions where there's stable coexistence of the two species. And then as we go to higher dilution rates, we get the fast grower winning. And of course, we can do this experiment for many different dilution rates to experimentally map out the bifurcation diagram associated with competition of these species. Right, so that's what's shown here. This is the fast grower fraction as a function of this dilution factor varying from, we actually went down to 3 up to 1 million. And in solid is the equilibrium of those two, starting at 0, coexistence, and then 1. And then in gray is the fractions that we measured where we started at different places and then we measured to see where they went. Right? So what you can see is that this really does have a simple kind of bifurcation diagram. And these are trends-critical bifurcations because this is an exchange of the stability of those fixed points. And again, we can just draw our simple subway map where it's the slow grower that survives down here and the fast grower that survives up here with a region of coexistence between them. So this basic simple expectation from the kind of model is, it seems to hold up, right? Here we cross the region of coexistence, whereas over here for another pair we're crossing the region of bistability. Okay, so again, over here the slow grower is outcompeting the fast grower. Eventually the fast grower wins, but here we cross a region of bistability where the winner depends upon where we start. Okay, but the bifurcation diagram here looks, so it's still a transcritical bifurcation, but now it's in the other direction because now it's an unstable fixed point that kind of crosses here. And this is then the subway map associated with this pair, where there's bistability at intermediate dilution factors. Yes? Okay, so the dotted line. Yeah. Oh, so we just drew in a line in between our experimental measurements. Well, so the gray is like telling us where we started and then where it went. So like here, we know that it's in between the two. Does that make sense? So we don't know exactly where that dashed line is, but we know that it's in between those two measurements. Oh yeah, indeed. And there are some cases where we know it's actually a saddle node. It's just, yeah, so because and also here, just like we were saying before, there could be coexistence at a low fraction that we can't measure, right? Yeah, so it's really, what we know is that there's an unstable fixed point, but this could come up a little bit, yeah, so it's only good to within 1%, is what I'd say. But in other cases, and we've done a lot of work probing fold bifurcations, and so they're my favorite kind of bifurcation. But there are some other cases where we know that it is. Yeah. So this is really just what you get from the simple model. It's consistent with our data in that sense, yeah. Okay, so these are some of the different behaviors of the pairs as we turn a simple experimental knob, right? And I have to say that this is very simple, but I'm kind of embarrassed to say that we've done a lot of competitions of species, and it was never obvious to me what the role of this dilution and like students would come into my office and they would say, oh, how should I do the experiment? Should I dilute by 100 or 1,000? And I'd be like, whatever, you can choose, right? And so I think, there wasn't really, I think, a clear thought on my part, but then also I'd say the fields of, how these things kind of couple within the model, but also just within, these simple experiments. Okay. And maybe I'll actually, for the sake of, all right. I already told you about these assembly rules. Okay. So this was what I showed you guys before. Right. So this is what we measure when we compete the trio. Right. Where there was this funny thing of exclusion, coexistence, bistability. Right. So kind of complicated. Right. But it turns out actually that all of this behavior can be made sense of just by the pairwise outcomes as we vary the dilution rate. Okay. So I hope that you guys can see this. So this is the three pairs where here it's what I showed you before. Slow grower wins, coexistence, fast grower. Here the two coexist throughout this region here until the fast grower wins. And then that third pair, we go from the slow to the fast winning, but in between there's a region of bistability. Okay. Right, and then you can just go and you can apply the assembly rules that I told you about before to kind of predict kind of what happens. All right, I'll mention, okay, we did this 8 times, and actually 2 of the 8, we saw that it went to a coexist, it went to this thing, so maybe it extends a little bit, but kind of at the edge of what you would maybe expect, so, so sometimes things don't quite work. But in general, I would say the pairs do a remarkable job of telling you what's going to happen in the trio. All right, so just to kind of walk through here a little bit. Okay, for example, on the far left, how do the assembly rules work? Well, what you can see is that the red and the purple can coexist. All right, that's great, except for the fact that each of them is outcompeted by the blue in pairwise competition. So the assembly rule says, well, maybe just the blue is going to survive, and indeed, that's what we saw. You come over here to the dilution factor of 100. Now the blue and the red can coexist, right? And that's useful because here the purple can't survive from the blue, so you'd expect that only the red and the blue coexist, and that's indeed what we see. At 10 to the 3, this is interesting because now you see a case where the bistability at the level of the pair propagates up to bistability at the level of the trio. Right? You can see that there's coexistence of each of these pairs, but bistability between purple and blue. So you'd say, oh, then purple and blue cannot coexist together in the trio. But it could plausibly be that you get coexistence of the other two at each there, and indeed that's what actually happens. The bistability propagates up from pairs up to trios. We've seen this up to 4 and 5 species as well, by the way. No. Blue and purple cannot coexist. So that's what the subway map here, it means that, in that condition, it's bistable. That's what, yeah. So this is, I would say, encouraging of a bottom-up approach to these communities, right? And even though, it might be a lot of work to measure these pairs, but in principle, it's possible. And I think this is telling us something conceptually about, how prevalent or strong these higher-order interactions might be. And I will just mention that you can take another trio, and in a totally different environment. This is now, instead of being a more defined media, this is a rich media. You can go and actually, the community assembly rules work here as well. Okay, so it's not just that particular pair and that particular media. It seems to be a pretty general thing. Okay? And then finally, I'll mention these community assembly rules also seem to work in the gut of the worm. Right, so we have a whole set of experiments where we're using C. elegans as a model host for a microbiome. What we can do is we can feed the bacteria to the worms, look to see which bacteria colonize, how they do, and we can ask, all right, well, what determines how well a species does, and so forth. What we find is that the trios can typically be predicted by the pairs, but they cannot be predicted by the single species colonization ability. Okay? So if you want to know what's going to happen in the 3 species, then you really need the pairwise competitive outcomes. Yeah. At the very beginning, all right, well, too late. No. Yeah. Right. Okay. So the one that's most okay. The question is, what would be a higher order interaction? And one would be, let's say I make a toxin that kills Madhav, so I can beat him in pairwise competition. But you make an enzyme that breaks down that toxin, and then he can now outcompete me because he's a fast grower. Right? Because you're fundamentally changing the nature of the interaction between us. It ends up not being, because it may be that you're not affected by the toxin at all. All right, so I think this is going to end up being a longer conversation. I think that maybe the general statement is that even without toxins, if you just write down a general resource explicit, you just write down a random model that you want to write down, in general, it will not actually be reducible to pairwise interaction. So that would be a special feature of a model. So generically, there's a sense that you should not expect it to work, but maybe it typically works or so. Yeah. Nope. Which makes it even more amazing. Yeah, no, I think that, yeah, so you can, indeed, if you look, there's a lot of spatial structure that you can actually see in here. And we've done, a variety of things. Yeah, but we find that actually the pairwise assembly rules also seem to work even when there's spatial structure. So I don't think that actually, the key thing is that you want to do the pairwise competition in the environment that you're interested in, whether it's spatial structure or temporal or whatever it is, and then it does a pretty good job of predicting what happens in that environment.
Part of one side of the pair.
Oh, yeah, and what we would call that coexistence, because if you get colonization here and colonization here, then they're both able to survive in the gut. Yeah, so that's yeah, and so the mechanism of coexistence could be spatial segregation, or it could be resource niche, separation of niches due to resources. So there are many different modes of it, but I would view spatial structure as another mode of generating, say, different niches. All right, with that, I think I will quit. So if I went there, maybe. Okay, perfect. All right, so so we've basically been trying to take this ultra-reductionist kind of phenomenological approach to the question of community assembly, at least in microbes. Right, so the reality is that everything is very complicated. Right, so they're all growing, dying, secreting lots of things, importing lots of things, and so. But I would say that the more complicated it is, maybe that's even a stronger argument for using a phenomenological approach of just some effective interaction between us, because it may be I'm secreting 20 things and Madhav is eating 10 of those, right? So then it's great if we can just reduce that to a single effective parameter. And what we find empirically is that these pairwise competitive outcomes can typically predict what happens in kind of like 3, 4, 5 species communities. We also did one with 20 species, and that worked there as well. Right, but sometimes it fails. Right, and this was an example of a failure of the simple prediction, but it could point us towards interesting molecular mechanism as well. What we find is that these pairwise competitive outcomes that capture just the effective result of the two species interacting, they change in some cases, at least in very predictable ways. At least for under changes in mortality, there are very predictable ways that these things change, and that's useful because we can combine the two results to start to say something about how multiple species these multi-species communities are going to change in response to some changing environments. All right, and with that, I'll thank the group who are the ones that have been doing the work. I'll mention, so Jonathan Friedman has led a lot of the work on community assembly. He now has his own group in Israel. I encourage people looking for postdocs, et cetera, to check him out. Nicole Vega was a postdoc who led the worm work. She now has her own group at Emory University. And Claire Abreu has been leading the effort on changes in the community structure in response to differences in mortality. With that, I'll take questions if you have any more.
Do you think there would be any advantage to higher order? These, as I understand, are not actually occurring communities. You might there could be advantages where those types of communities that are unusual in this sense.
Yeah.
Be selected for.
Exactly. Yeah. So I think that, right. So one thing, so this is all basically random species I mixed together. So you can think about this as de novo community assembly, like dust blows in from elsewhere, and then now these species are in some environment. What happens? Also, you can look at the network that we had, where, for example, we don't see any non-transitive rock, paper, scissors interactions in that network I showed you. But again, these are not natural communities. So maybe if you go out and you look at a natural community, things will be very different, right? So to at least take a step in that direction, what we did is we went outside and we grabbed a single grain of soil from the front yard that was kind of 10 centimeters down. And from that single grain, we isolated 20 different species. And then we did all the pairwise competitions of those 20 species. And what we found is there were still no rock, paper, scissors trios. Right? Over 1,000 triangles we looked at. Couldn't find any. We went to a different media. Couldn't find any in the other media. Right? And then we mixed all 20 species together. And what we ended up getting was 3 species coexisting that were exactly the 3 species that you would have expected from the community assembly rule. Right? So it's, okay, I'll let you decide what that means. Yeah, and so it's a mystery. What's going on, right? And so we have definitely not solved the problem of the diversity in nature. There's a tremendous amount of diversity everywhere you look, yet it doesn't seem that the mechanisms that we have at our disposal are quite enough to generate the diversity that's actually observed. So, so I don't know. Yeah.
The thing that was puzzled, that segregation, educational bias than local bias rather than mixed. So obeying something different.
Well, okay, so in the gut of the worm, it turns out that the initial colonization event is actually a relatively rare event, and that's what's leading to the spatial co-localization that you were seeing in that image. So that's one. Yeah. So it's not actually local. It's not biostability. And then and then the question of additivity. So and I think that there are many things to say. One thing is that this is all maybe just a simple approximation. I mean, the realities, of course, is that it is nonlinear and it is not additive, right? And so the statement is that these nonlinear effects are not so large as to just throw everything off. So I think that's maybe one way of thinking about it. We're also very interested in this question of what are the signs of these interactions? What's the distribution? Because we've focused a lot on questions of cooperative growth and how that leads to to these fold bifurcations and tipping points and so forth. I can say is that most of the interactions that we see are competitive, and sometimes we see a one-way one-way facilitation, right? And in particular, if you are a crappy grower in a given environment, then when you have a competitor, it's likely going to wipe you out. But if it doesn't wipe you out, it might help you, right? Because it means that you're in an environment where you're missing something, and so you can maybe benefit from some secreted product. So that's really the situation where we see benefits, is that a crappy grower can sometimes benefit from having a competitor. But in general, it's competitive. And we see essentially no examples of two-way mutualisms in our data. Yes? All right, so you're wondering, for example, of what sort of food sources would facilitate one or the other outcome? Is that okay? Yeah. So I basically haven't said anything about the media here. We're very interested in this question of what facilitates coexistence or not. One thing I can say is that we frequently get coexistence on single limiting carbon sources. Right. So this whole idea that the number of species has to be less than the number of limiting resources. I mean, it's true in a model, but then you have to remember that a model is making some set of assumptions that may or may not be true. And in particular here, I think there's often creation of additional nutrients. Right? So even if you have just glucose, it's important, but then you get secretion of a bunch of other things. So those are in principle new niches. So we see, as a function of the complexity of the environment, it does facilitate coexistence a little bit, but not as much as you might think. Quit? Yeah. But I'm happy to take more questions. Yeah. All right, thank you.